Hello everyone, my name is Akanksha Kare and I am a graduate student in Vaccine Nanotechnology Lab at Mercer University College of Pharmacy. I will be presenting my poster titled Zika Vaccine Microparticles Loaded Dissolving Microneedles Induce a Significant Humoral Immune Response. Before I go over the results, let me give you some background about the disease. Zika is an infectious disease caused by Zika virus and transmitted to humans through mosquito bites. The detrimental effects associated with this infection highlight the need for a safe and effective vaccine for Zika. Hence, the purpose of this study is to formulate and evaluate vaccine microparticles encapsulating inactivated Zika virus as the antigen. Subsequently, the vaccine microparticles are loaded into dissolving microneedles and administered via the transdermal route to provide painless and needle-free immunization. There were four objectives of this study, which I will discuss during the methods and results section. The vaccine microparticles were formulated using double immersion solvent evaporation method using PLGA as the polymer. These particles were lyophilized to obtain the dry powder which was further loaded into dissolving microneedles. The microparticles were characterized for size, zeta potential, morphology, antigen integrity, in vitro release, in vitro immunogenicity, and cytotoxicity. These vaccine microparticles were administered to mice in 3 doses, 1 prime and 2 booster doses. Serum samples were collected to monitor the antibody titers. Now let's look at the results. Particle size of these vaccine microparticles was found to be 573 nanometers with a PDI of 0.294. This indicated a narrow size distribution. The zeta potential was minus 22 millivolt with an encapsulation efficiency of 50 to 70%. The average length of microneedles was 400 micrometers. As you can see in the picture, the microparticles were spherical in shape and the microneedles dissolved in 10 minutes after application into mice skin. STS page showed that antigen extracted from microparticles produced a band at similar molecular weight as that of the antigen in solution form. This confirmed that the formulation process did not affect antigen integrity. In vitro release study showed that 50% of the encapsulated antigen is released within 24 hours, while remaining 20% is released over a period of 1 week, indicating the sustained release characteristic of our vaccine microparticles. Crisis assay showed that the cells treated with adjuvanted vaccine microparticles produced significantly higher amount of nitric oxide, which is an innate immunity marker. Literature had showed a link between antigen presentation and autophagy. Hence, we evaluated the ability of our vaccine microparticles to induce autophagosomes in murine dendritic cells, and the cells treated with vaccine microparticles produced 20-fold more autophagosomes when compared to the cells treated with solution or blank microparticles. MTT assay showed that the cells treated with vaccine microparticles at different concentrations were viable as compared to the cells treated with a known cytotoxic agent, that is TMSO. The serum sample of immunized mice showed that mice immunized with vaccine solution, vaccine microparticles via intramuscular route, and adjuvanted vaccine microparticles via transdermal route produced significantly higher antibody titers. These serum samples also showed presence of antibody subtypes, which are IgG1 and IgG2A. The antibody titers were comparable between the intramuscular route as well as the transdermal route. To conclude, I would like to say that Zika vaccine microparticles with sustained release properties were formulated. These particles were safe and immunogenic in vitro. They elicited significant humoral immune response after intramuscular as well as transdermal immunization. Humoral immune response after transdermal administration was comparable to immune response after IM administration as seen from the antibody graphs. Induction of IgG2A and IgG1 antibodies showed that the vaccine microparticles was able to induce both Th1 and Th2 mediated immune response. Thus, the feasibility of a potential transdermal Zika vaccine was established using this study. Future studies will comprise the evaluation of cell-mediated immune response and memory immune response induced by the Zika vaccine microparticles. These are some of my references, and I thank College of Pharmacy for allowing me to conduct this research. And I thank you all for your time.